Hi, I'm Brad Power. I'm the host of the Prostate Cancer Lab meeting. Today we have 3 items on the agenda. We want to update you on a conversation that was triggered out of our last meeting, which was right to see, the idea that patients should have access to research use only data that is run on them. Second is Rick is going to lead us through his treatment situation, his history, and the decisions that he's facing now as a kind of a setup for people in general with advanced prostate cancer. And then also he's going to share the data that we have on him and Brian McCluskey and talk about analyses that he'd like to see run. So that's our agenda for the day. Let me just update you on the conversation quickly that we had with Nick Shork, Peter Kuhn, in particular, Marty Tenenbaum. And what they suggested, there are varieties of ways to get around the roadblock on patients getting access to research use only data. One is to have a workaround with an IRB that Nick Shork talked about, that Rick has gotten. Another is to broaden that and expand that, and that's what we're talking about. And apparently we can set up an observational feasibility trial, which lowers the barrier on like a classic randomized clinical trial, which is prospective and is very expensive and slow. So we're working on that, trying to find principal investigators and or organizations that would fund such a research activity. So if you know anyone who's a PI, potentially a PI or a research organization that would want to fund that kind of work, let us know. Anything on that, Rick? And of course, the other front is to go through the regular channel. So Rick's trying through the City of Hope to get his tissue through the tests and the machines that they have that would be CLIA certified or that would be accessible to his oncologist. Anything to say on that front, Rick, about the general right to see for your for these advanced tests?
Not specifically. It seems like things, but generally, it seems like TGen is moving ahead with getting approvals to work with Akoya Enable. The reason I can't say specifically is Akoya Enable doesn't want to talk to a patient. They want to talk to TGen, who has the protocol in place. So I'm kind of boxed out. I get a little update that things are moving ahead, and I'm very excited about that because I would like to be the first of many. And I'm really interested with what Rick Davis is going to say, because we had such a good conversation last week. But in general, I'd like to see this available to everyone who has the means. I think it's pretty modest, a few $1,000s. So just a generality, I think it's getting closer, but I don't know specifically.
Okay.
I wouldn't mind inviting a little introduction for a few people that I don't know and that I'd like to welcome. If I don't know Chandra or Jan or H. Messerly, if you guys wanted to just say a quick hello, I thought it was very interesting to get the little background from Saeed and Alex. So if you guys wouldn't mind just saying a quick hello.
Sure, I'll go first. I'm Chandra Kota. I'm what you call is a medical physicist. I don't know if any of you have had radiation treatments before, you would know.
Yes.
I'm currently working at Yale. I'm interested in looking beyond just my immediate job, day-to-day job, and see how I can help patients out with maybe some of the newer radiation treatments and ideas coming out of the research field.
Beautiful.
Heather.
Hi, I'm Heather Mesterly. I'm at Certus Oncology in San Diego, Sorrento Valley. We work with mice creating OPDX models of patient cancer tumors and then test different treatments and therapies on them to see which work and do not work. I'm not a scientist, I'm the patient manager here, and I'm learning so much by sitting in on this, but I'm just if anything pops up that it seems like Certus might be able to step in and help, I'll definitely let you know. And if you're in the San Diego area and you want to visit our facility, you can.
Oh,
Thanks,
perfect.
Heather.
I used
So
to
that's
work at
one of,
longevity.
and that's one of many. I'm sorry, that just what Certus represents is one of many kind of vectors that we're exploring or work streams, and that's the organoid and testing. And so Certus offers traditionally mouse models. Right. So and Brian is in the San Diego area, so we're hoping that we can have that conversation and see if it would be suitable, because both Rick and Brian and I think other patients, you know, they sort of go I don't mean to say this badly, but they bounce between treatments. And so they're constantly in the mode of testing, and so they have the ability to take a long lead time in terms of getting a testing environment set up for themselves, because they're going to be coming back to decisions repeatedly.
Yes.
So I think the profile of the prostate cancer patient fits for the Certus services.
Yes.
Okay. And Jan, you want to introduce yourself?
Well, I've been here before. I think I've met Rick, but yeah, I'm a patient. I'm just a patient. I'm not a scientist. I have castration-sensitive prostate cancer still, so I'm lucky in that way. I just inquired with my doctor as to whether I could get the lutetium-177. They said nay, because I have to be castration resistant, and I have to have the Taxol treatment, and basically I have to be further along in my journey, I guess, before I can access that technology. So anyway, that's about it.
Thank you.
Yeah.
I think we'll get into that. I think Rick will tee that up. Rick, we have two Ricks. I think Rick Stan will tee that up, and I expect Rick Davis to comment as well on all these the treatment options and the considerations of prostate.
I'm going to turn off my video because I'm eating and stuff, so I didn't want to.
Yeah. Okay. I think he's in the Seattle area, so I understand when Laura's having lunch, but it's 9 a.m., so I don't know, maybe late breakfast. Okay. All right. So next topic, Rick, take it away. I assume you want to start with, there were two topics. One is treatment, and then the other is the data that we have that would inform that treatment. So I assume you're going to, lead us through that conversation. And I would encourage everyone to, interrupt with questions as we go to make it more conversational. Thanks.
Yes, thanks. Thanks, everyone. I thought I'd start off with how am I doing? It's always, when I was on other hackathons, it was always interesting. How is the patient doing? Well, I had some good news this week, and I always like to share good news. I think it's a little crazy that I get my PSA blood work done at the same time I get my docetaxel infusion, especially when you're near the end of your rounds. I just completed 6 rounds with, and I got a 80% infusion because I started getting tingling in my fingers. I am a guitarist, and I want to live more than I want to play guitar, but I guess it's standard protocol. If you report you have you're starting neuropathy, they immediately cut you to 80%, and that's just how it works, I guess. I don't know. That's what happened to me. So I then get my infusion, and I wait on pins and needles and hope to find out that my PSA went down, which it did. Not a lot, but it went down. So I'm at 2.4. Last time was 2.6. The time before was 2.4. So I feel like I'm just bouncing around in noise around 2.4, 2.5. So I'm still, to my awareness, being controlled. And so I have another Chemo's planned, even though I'm at 6, and at the start of the chemo, I was advised I would be going 6 to 10. So I think my doctor is Dr. Lou, Sandy Lou. She's the head of a clinical trial at UCLA, ARCUS 6. ARCUS is my good pals. I used to work with their management at Amgen, and they started a company called ARCUS. Very good small molecule company. And I'm on the clinical trial, which is docetaxel, a PD-1 inhibitor and an adenosine inhibitor. And I had hoped to get on that trial arm, but unfortunately, I just got on the docetaxel only because it was not double blind, but it was randomized, and I just didn't get the luck of the draw. So I need to make a altered treatment decision upcoming, and I'm happy to say I don't have as much pressure. If my PSA was going up and the scans were showing growth, I'd be under time pressure. But now it looks like I have a little time. So that's my update. I'm pretty happy to tell you that. Any questions? Did I cover that okay?
Dr. Laird, anything that you would, as an MD coming fresh to this, anything you would have questions about as a background?
Just the extent of the disease, where is it located, and what was previous treatment?
Sure. I'd love to cover that in a moment, because I've tried to develop a one slide or somehow succinctly communicate my state. And so you will be the new guinea pig. I've communicated this to my 3 medical oncologists, and they all said, well, it's okay. And when I saw Brian's depiction of his state, which was a PSA chart in his journey of interventions and PSA, I saw how much more lively that was. But here's my state. But kind of like an outline of what I'm hoping to go through, just to kind of go through it as my decision, my treatment guidelines down the NCCN decision tree, and how I'm reaching the end of that decision tree, and how I would desire my maybe upcoming therapy options to be influenced by me personally, my cancer state, immune, genomic, and patient state. And so I'm hoping it's going to be informed by an IHC at a minimum of something like we're seeing on the right here, where these are 4 tumor slices. The upper left, and this is the brown is a CD3 stain. So that's T cells, the combination of CD4, CD8, and Tregs. And you can see abundance in the upper left of brown. So this tumor is what would be called inflamed or hot, or loaded with TILs, or tumor infiltrating lymphocytes. On the right, you see it's cold. There's no CD3, T cells are not infiltrating this tumor. On the lower left, you would see that if you did an RNA-seq, you would think that you have good infiltration, but you can see that I'd call it the moat of exclusion. There's an immunosuppression that the T cells are unable to cross this boundary. And in the lower right, you can see basically a very low tumor infiltrating lymphocytes, but they're there. The reason I bring this up, these 4 states, is they could, to any researcher who understands how T cells work, this would influence your selection of immunotherapy. Either you need to do if you're here, you need to do a prerequisite, or just hope for a miracle that you'd be one of the few people that respond with no infiltration. So anyway, how did that go? Any questions on that? And
Which do you look like?
I don't know. So I'm about to share my state to my awareness. I'm waiting on IHC, which stands for immunohistochemistry, which is this stain. It's an immunohistochemistry stain where a pathologist will add a stain which will bind selectively to whatever they are looking for. In this case, it's CD3. So I'm waiting on that from City of Hope. It was requested by Tanya Dorf, which was a beautiful thing. I'm on the phone with her and I said, well, could I just get an IHC done? Because this is a subset of what we're seeking to do here at the hackathon and open up for other patients. This is what I'd call spatial analysis. And Akoya, Enable, NanoString all do very deep queries, and they're not mainstream and they're research use only, but I'm hoping to do what is standard IHC and compare it with some of these other technologies to make sure they all kind of say the same thing. So I certainly hope, and any patient would hope, that they're in the upper left, or at least the lower right. You want to have, if you have no, like if you're in the upper right and you're cold and you have no tumor infiltrating lymphocytes, to human awareness, common like a PD-L1 blockade inhibition is not going to work because there's no T cells to modulate here. So let me get back to my next slide, which is going to be a dense slide, but I'm hoping that it'll communicate where I'm at. And
Rick, could I make a comment on that?
please, yes, please.
So thank you for that. Your upper right-hand slide to give immune therapy is probably a waste, as you probably know. It's not likely to be beneficial. So prepping that microenvironment is really a key. How can that be shifted? It's always a key issue. There's a group, they're not sort of set up commercially, but there's a group called Consultative Proteomics at the University of Texas, Dr. Robert Brown, that I've used. They do basically, it's proteomics, so they're doing IHC stains, 20 or 30 of them on any given tumor. I don't know if you guys have familiar with their work at all.
No, that's beautiful.
What they do in terms of your particular slide that's directly relevant is they will assess CD8 cells, which you might say are the ones that are going to be most helpful.
Right.
FOXP3 cells, which are put on the brakes. And so they look at the number of CD8 cells per high power field, FOXP3 cells per high power field. Anything 2 to 1 or over is considered generally pretty good. And then they'll do CD163s, which are macrophages, which are also key. And macrophages can be in a M1 stage or an M2 stage. And if they're on the M1 part of and they transform back and forth. So if they're in the M1 phase, then that's very positive. If they're transformed to M2, then that's another break on the system. So that's the most useful thing I found for kind of commercially available, but they're, we can get them, they'll run samples. They're just not set up to be a high volume lab, and sometimes it takes 6 weeks to or more to get the results back. But they're very helpful. They're, interested in these kind of things. And this look at the microenvironment was something that my mentor, Mark Renneker, actually had them set up a few years ago. So they also have, you get a protein fingerprint, which can give one a sense of which of these genetic changes are actually driving the tumor and which aren't. No oncologists are going to make any treatment decisions based on this, but in terms of looking at the microenvironment terrain, there often are off-label products that can be used to help in the microenvironment.
That's beautiful, John. That's so insightful. I feel like, okay, we're ringing the bell here. So I feel like going 20 deep in IHC. Do you need 20 different slices of tissue? Or are you able to stain and wash?
I think they end up with 20 slides. If they can get a hold of the block, they can make a lot thinner slides than the typical path labs. But offline, glad to help you get set up on that.
Are you familiar with Akoya and NanoString's spatial efforts?
I'm not.
You're gonna love them. So it's immunofluorescence. It's just like an IHC. You're able to do it on one slice of tissue. Immunofluorescence allows stain and wash. So you're tagging a fluorescent molecule, typically red, green, blue, some color, to an antibody. And you can go in 3 at a time and resolve the 3 colors. And so you would pick, let's say, CD3, CD4, CD8, Fox, just keep going, because you can dissociate, wash away, and go in with another 3. So they can iteratively build up to very high dimensional. I've heard like 100 plex. I know they're operating at 35. This is both, this is Akoya and Enable. Interesting, James Allison is on the board at Enable. So you'll love these guys. It's exactly what you said, except for it's on one slice of tissue. So you have the exact same slice, and you're able to query very deeply. And then NanoString has a different offering also for even deeper querying of basically intersections of where, let's say in this slide, a little lower left, you would look at the boundary between the tumor, which is in the upper left, and the immune cells that are trying to get into this tumor. So you would query selective boundary areas with the NanoString. So yeah, I think you're going to love it, and more on that to come.
And Brad, that'll be in the notes.
Yeah, basically I transcribe all this and try to provide links. So
Okay,
you just mentioned the company, which went in one ear and out the other, but when I review the recording, I'll try to put a link to them in the notes.
Yeah, you'll love them. Okay, so here's me. I've presented this slide to Dr. Raina McKay, Dr. at UC San Diego, and Dr. Tanya Dorf at City of Hope. So I try to summarize. It's a dense slide, so I'll just kind of go over it kind of quickly, because there are some things that are kind of interesting. So from the row 20 to the bottom is my PSA. In 2019, just at a yearly physical, I went from like 3.1 to 4.1, And that's when I found out, basically that triggered. So I've had my prostate out. So you can see my treatment history. I've started Lupron, Casodex. That held me stable for 8 months. Then darolutamide didn't do much. Now I'm on docetaxel, and you can see me bouncing around. I came from 3.6 down to 2.4. Okay, so that's that. And then I desire to be aggressive. So far, I'm fairly healthy. And if I look at my treatments, I've had salvage radiation, Lupron, Casodex, darolutamide, and now docetaxel. Okay, now, so what do I do next? And this is where it gets more interesting.
Rick, I think Dr. Laird was asking about the extent. So you're metastatic. Can you talk about?
Yeah. Yep. A nodal disease, 4 lymph nodes from my neck to my pelvis, light up in a PSMA scan and are confirmed in CT scan or MRI, or I'm not sure. So I'm at nodal stage 4, castrate resistant. Did that answer the question?
Yes, thanks.
Sure. So this lines 3 through 9 are my asking of my next therapy options. So when I talked to Tanya Dorff, she noted, and so this is like what evidence would influence her recommendation. So here you can see in line 3, the CDK12 mutation is my driving mutation. And that is going to open up a lot of fusion neoantigens by nature. And so she is advocating this clinical trial that combines bispecific PD-1, CTLA-4 with olaparib. Okay, olaparib is an interesting recommendation. It has been approved for CDK12 mutations, but subsequent data shows maybe not that great. More for a BRCA 1 or 2, that would be shown some good efficacy, but olaparib relative to CDK12, not so much. So then I asked her, well, why would you recommend this trial? And a big shout out to Rick Davis, because, our discussion last week, totally changed this actually, and I'll get to that in a moment. But she said, well, olaparib on its own as a single agent probably wouldn't do anything, but in combination with the PD-1 and CTLA-4, think it might have a good fit for me. So I said, oh, okay. So then we move on. So that is what one of the leading medical oncologists recommends for me coming up after chemo. Okay, then Dr. Raina McKay she came up with several to consider, with her top one being a PSMA CAR T. So that sounds pretty good. I don't really know that much about it, but why would she say PSMA? Well, there's 2 evidence notes that why it'd be a fit for me. Population studies would be the first, just like metastatic prostate cancer patient guys like me tend to have a high expression of PSMA. Okay, so that's population study. It was also confirmed in the RNA-seq. So I'm super high expressor, so is Brian, of PSMA. And it was also confirmed by the PSMA scan. So we know that lights up. So anything with a PSMA target is a triple fit for me, from just population studies to the scan to RNA-seq. Okay, this seems to make sense. Another one.
Rick, Dr. Laird has a question.
Sure.
Thank you. It's actually a suggestion. Have you worked, and is this group familiar with the work of what's his name, Robert Nagorny down in Long Beach?
No.
So sort of the bottom line is Bob Nagorny in the 90s started doing chemosensitivity testing, which is basically taking live tumor and exposing it in the test tube to multiple different chemotherapeutic agents. There are many other people doing the work at the same time. The science was crappy. The whole field got discredited, but Nagorny and a couple other people done excellent work over decades. And in the test tube, you can test different combinations to see what's going to work. In general, if he finds that a tumor is sensitive, that will be borne out 80% of the time clinically. If he finds that any particular chemo is ineffective, that's borne out clinically 90% of the time. Now, Nagorny is one of the smartest oncologists. In a room of oncologists, of 100 oncologists, he could most often be the smartest one. I mean, he really looks at this stuff deep. He's world class. He's had to, he's got a tough ego. He's taken a lot of hits from the profession, but it turns out some of his work has actually led to standard of care combinations, gemcitabine carbo and carbotaxel, carboplatin, excuse me. What the larger point that I'm getting to is that trials are a crapshoot, and the percentage of success, you guys probably know better than I do, but my kind of sense is, maybe 15% would be pretty good. Maybe some trials are higher, but it's kind of a crapshoot. So my sense is it would be rational for my patients, and I want to say I usually don't give any advice to anybody until I've looked into their case for 4 or 5 hours, and I don't really know your case or what's going on, but I have the general sense. And this is more of a wide open conversation kind of format. To me, it makes sense to go the Nagorny route before you go into trials, because you're looking at 80% or 90% accurate, clinically useful information as opposed to 15%. So I would go that route first and get his assessment. The larger issue here is he doesn't care so much about genetics. I mean, he'll look at it, he'll look at the proteomics. He looks deeply into what are the mechanisms of this cancer, but he's sort of like Harrison Ford, when that scary Arab guy shows up in the market and he's waving all of his swords and stuff like that. He just pulls out his gun and shoots them. Nagorny cares, will the treatments kill the cancer? So he sort of cuts to the chase kind of guy.
Well,
I think you like him. This guy he's great to have on your team. If you've got nodal disease, then it's highly likely that a centimeter of fresh tissue, which he needs to get to his lab the following day, but a centimeter of fresh tissue is probably readily accessible. Some patients just can't access it. It's hard to get. So whereas the proteomics is stored tissue, Nagorny needs fresh tissue. But I just sort of insert that as before you go down the trials avenue. Consultation with him is definitely worth doing.
that sounds great. did I spell his name? Nagorny, Nagorny?
Nagourney, Nagorny Cancer Institute. He's got an old, what do you call it? He's got an old talk, TED Talk. He's got an old TED Talk on his website, but you get a sort of a flavor for him.
Thank you
And he
very
can
much.
consult with oncologists. I mean, he's not just a lab, he's an active practicing oncologist. So he knows the challenges, and you could probably go down to his setup there, and he's got surgeons who, he's worked with for a long, long time. They can work it really smoothly, or you can, we can send tissue to him anywhere in the world as long as it gets there the next morning. But to do it on site does have an advantage. You're more likely to get the cells as viable as possible.
Rick Stanton, I want to give Rick Davis time to talk. I don't want to wait until the end of
Yes.
the last time. And we've only got 15 more minutes.
Yes.
So I would ask you to focus on requests, things that you would want from the audience that we'll put in the notes, and we'll go out to the people who aren't here today. So if you can frame this more in not so much information, maybe you can put some of this up somewhere, but rather around requests you would have, places you want to go, questions you have.
Okay. I feel like I would like to invite Rick Davis to talk sooner than later as well, because he was so influential in my awareness and decision-making during the week. So I can frame this, Rick, with these were the recommendations by Tanya Dorff and Raina McKay. When you gave that beautiful talk or suggestions about Pluvicto, like, hello, why wouldn't you be doing something that has, some evidence behind it? It was like shocking because it didn't come up in my 3 out of 3 clinical oncologists. So I queried all of them during the week. I got a response, a beautiful response from Dr. Sandy Liu, who's my current treating one under the docetaxel and the ARCUS trial. Her response was, yes, Pluvicto would be a very good option for you. So that is so interesting, because until I brought it up, because you brought it up, wasn't even on the radar. And suddenly it's a very good option. The other option is a crossover that I can actually go to. I can stay on docetaxel with the PD-1 inhibitor and the adenosine. So I wanted to say thank you, Rick, because I believe Raina McKay also, but I got to look at her response specifically. But basically, you fleshed out something that that was great. So please weigh in here, Rick, because I wanted to say thank you for.
It's a pleasure. It's what we do. Sometimes you can't see the wood for the trees, and I also want to follow up and thank Dr. Laird, because I think he's saying the same thing. We can be looking around and running around in circles in the spiral staircase up the ivory tower, and there's some really good stuff that's out there. I don't know. I put Nagorny's link in the chat window so you can go straight to it. I don't know how much experience Dr. Nagorny has with prostate cancer, but it's an unusual, it doesn't behave exactly like everybody else, like other cancers. I'm just looking at this chart, and something that Dr. Laird said and suggested Nagorny actually came to me simultaneously. Your disease was strange from the first place, because it's been very aggressive, but it only showed up as 4+3. And when I say only, 4+3 is relatively aggressive, but it didn't have a Gleason over 8. So I am thinking and wondering whether your slides ever got reread, whether Epstein at Johns Hopkins ever looked at them, and whether in fact your disease was a much higher Gleason, which would explain right now, and if it was possibly introductal disease, which a lot of lesser pathologists don't identify unless you actually ask them to identify it. Now, why would that be important now? Why do we need to know that now? Well, if you had higher Gleason, if you had introductal disease, and you had very low PSA, then there's reason to think that from the outset, your disease didn't really behave like an adenocarcinomic disease. And there may have been mutations in there from the very beginning, which didn't allow you to respond properly. Now, one of the treatments that we've seen used in that situation is adding a platinum-based chemotherapy. And I think you're just getting docetaxel right now. And I would be doing a little more work to look at those cells and see, well, did they look like small cell? Did they have like a neuroendocrine aspect to them originally, or even now? And if they did, maybe we should be adding something to the chemo. Now, this is totally separate from the PSMA discussion. But, you know, when we go to these docs who are supposed to be so good and they don't see the obvious. And sometimes it makes me a little crazy. Were you diagnosed fully in the first place? Did we have all the information in the first place that we needed? When guys come into our group, these are the sort of things we're pushing them to do, to go back to their medical team and to say, have you done this? Have you done that? Did you send the slides to Epstein? There's nobody better than Epstein to look at those slides in the country, in the world. The only other thing I wanted to say is, I mean, I'm really glad that we've got you and hopefully Brian, if we can figure out the right way, onto the PSMA path. I mean, when you say you're super sensitive to PSMA treatments, well, the place to start is clearly with Pluvicto. I mean, we've got another gent who we hooked into Bryce yesterday, who is about to start, and his disease is running crazy. His tumor burden right now is around 39. So, I mean, it's going nuts. And he's certainly eligible for Pluvicto, but he doesn't want to wait to start the 4 to 12 weeks it could take. I'd be very interested to hear, by the way, from Dr. Liu when she thinks Calais is going to be able to start treating patients. That's the question we'd like to know. But he's going into the Weill Cornell trial with alpha plus beta particles. And he's starting that next week because time is so much of the essence for him. Fortunately, you don't have that situation. And if it were me, would I go into an alpha plus beta versus Pluvicto? Probably not. Oliver Sartor seems to think that there's potential in that based on what he said in that 6-7 minute video. I think it's valid, the alpha plus beta approach. But frankly, I'd start with what's tried and tested. The last thing I just wanted to say about the olaparib, again, you know, these doctors are just fishing around to see how they can make a cold tumor hot. And the only reason they want, and you've got CD12, so they, oh, olaparib. The problem with olaparib was that in that was it Profound? I forget the name of the trial, but in that trial that Maho Hussein led, they went for a basket approach of approval, and they got all these mutations approved by the FDA for the use of olaparib. But what they didn't do is show them which of those mutations were effective and which were not effective. They applied for a basket and they got a basket approval. But in fact, the only mutations that were sensitive to the olaparib were BRCA1, BRCA2, and maybe, I think with about 7% of people, ATM. And the whole rest of them, they were not. And now you got people who are all excited because they've got, you know, a PALB or an AT or a CDK12 or something like that. They think, oh, olaparib's the magic bullet. No, it isn't. And you know, we have to tie these guys back, and we have to stop getting these docs to view some of our folks as fresh meat, because that's driving me a little nuts. So I'll get off my soapbox right now.
I think it's a beautiful soapbox, and I want to mention thank you to all, because you mentioned something about your friend who he's out of time, you know, and I am happy, very happy to say that I'm currently, you know, whatever treatment, I'm suboptimal, I'm out, you know, at least I'm under control for today, and that gives us a chance to be proactive rather than reactive. I mean, when you're in a reactive mode, when your parachute fails and you're falling, it's like you don't even have that extra week or two to get the paperwork done, to get scheduled, let alone figure out what to do.
Exactly.
So thank you to all of you so much. I will spend 1 minute, and only 1 minute, so on the slide, because it's kind of interesting that I've been very lucky to have TGen, Translational Genomics, help me with Nick Shork. So we see these possibilities for my next treatment. And again, thank you, Rick, because Pluvicto is now on there. And then I wanted to mention the analysis that has happened for me, luckily, because of TGen and what I'm trying to do moving ahead. So from whole, I actually had whole genome sequencing done, not only whole exome. So we have the tumor burden, we see the fusion, we see the mutation. Also done on me was a neoantigen analysis. So similar to Shirley Pepke, my buddy Guang Fa at TGen gave a beautiful neoantigen analysis of my tumor, which is plentiful because of the fusions. And I have a lot of fusions. Then also TGen was able to identify the overexpression. Rick, you mentioned that I feel that I'll be sensitive to a PSMA. I don't know that, but I do know that I express PSMA highly, so that's encouraging. There's a connectivity map going on for me. There is an organoid study going on for me. So along the lines of testing therapeutics on organoids, that's happening. So it's just very nice analysis that TGen has brought to the table. Here's the last thing that I was surprised happily that Raina McKay said, hey, how about a B7H3 clinical trial? And we know that us prostate cancer guys tend to express B7H3. So that was the first time I had heard a doctor suggest a clinical trial based on RNA-seq data. So that's kind of interesting. So it's time for me to be quiet. We're almost out of time. Thank you.
So Rick, let me just bottom line this on this last section you just talked about, the analyses you have. So this is data that you have that you could make available or is in process and could be in a short while, hopefully be made available. If we were running a classic bioinformatics hackathon like Alex Feltus knows, how could do we need to schedule a separate event to do that? When could we do that? How would you like to approach that? Would you make it available now? How, like, sort of, what analysis do you want to do on top of the data that you have?
Right now I'm struggling. So I'm a ex-bioinformatician. So I am looking at my RNA-seq data to do an assessment of my immune composition using libraries in R. First off, this is my data. It's available on a Google Drive, and anyone who wants it can see my, mutational reports, my clinical notes, my HLA types, RNA-seq and transcripts per million from both ASIAN and Tempus, and my Tempus report. So this data is available for anyone who wants to hack on it. To answer your question, Brad, RNA-seq, by next week, I hope to share, my immune composition, which is a poor man's view to answer this kind of question, like, but are there Tregs? Are there CD8s?
Yes.
So you can estimate that rather exquisitely, supposedly, with this immune deconvolution. And if anyone wanted to help on that, that'd be great.
One thing here, actually, as I said, there are tons of data on the public domain, and we should learn from those data sets. And I have something to present today, but it seems we don't have enough time today. But I think there are a lot of things, for example, for drug repurposing, there are tons of data, and we should be able to use those data. I have one, I've done one, but hopefully I can present it next week or so.
Let's do that. Let's do that.
That'd be great.
Out of respect for everyone's time, I'd like to wrap now and stop the recording.